Good morning, esteemed speaker, Dr. Brijesh Rathi, participants, colleagues, and dear students and volunteers. Welcome to the last session of the National Workshop on Bioinformatics, Tools, Resources, and its Applications. On behalf of the organizing committee, I'm happy to welcome our speaker, Dr. Brijesh Rathi. He shall be talking on computer-aided drug design. What can be more apt than today's topic to culminate the workshop on bioinformatics? We have covered almost a large number of tools and resources and a few applications as well. In present-day context, when we are grappling with COVID-19 disease, treatment and prevention, I think the two major buzzwords are structure of the receptor molecule, efficacy of the vaccine, and the names like remdesivir and many other drugs that are being used for the treatment of the disease. Under the circumstances, today's lecture will help us understand how can we use the field of bioinformatics to design drugs utilizing the available digital data. I therefore feel this session is going to be extremely rewarding. Without any further delay, I take this opportunity to introduce Dr. Brijesh Rathi to the participants. Dr. Brijesh Rathi obtained PhD degree in chemistry from University of Delhi and is currently working as assistant professor of chemistry at Hansraj College, University of Delhi. He is a recipient of a prestigious Young Scientist Fellowship, Early Career Research Award, CAPE Sprint Fellowship. This CAPE Sprint Fellowship is from Ministry of Education, Brazil, and the prestigious Excellence Awards for In-Service Teachers by University of Delhi. Dr. Rathi has been visiting assistant professor at Department of Chemistry, MIT Cambridge, USA, and is presently affiliated faculty at University of Loyola, Chicago, and University of Debrecen, Hungary. He has published more than 75 papers in various journals of national and international repute. Dr. Rathi holds the position of section editor for current topics in medicinal chemistry and associate editor membership of chemical biology letters. Dr. Rathi has supervised 5 undergraduate research students in the past and is currently supervising 4 doctoral students and 2 master's theses. His main research interests are centered at the development of new antiparasitic and antiviral agents and their target identification. A highly decorated scientist and a lecturer par excellence. I now invite Dr. Rathi to deliver his talk. Over to you, Dr. Rathi.
Thank you, ma'am. Thank you so much for giving me this excellent opportunity to be part of this very important national workshop on bioinformatics that is organized by the Department of Botany, Zakir Hussain. So I congratulate all of you and thank you once again. So I hope, let me try if I am able to share my slides. I hope slides are visible.
Yes, they are visible. Thank you.
Thank you so much. So let me begin with again great thanks to all the colleagues from Zakir Hussain Department of Botany And I really congratulate to them for to organize this very fantastic bioinformatics, its applications towards drug discovery and other emerging areas. So, of course, I'm not a bioinformatician, I'm a medicinal chemist, but I'll be uh, trying to integrate all my expertise to the area of this, to the scope, within the scope of this particular workshop. So the title of my talk is Computational Supported Drug Discovery. So let me be very honest that we were not working in this area before this COVID-19. So the COVID-19 during that pandemic, we all were puzzled, and everything was kind of collapsed globally. Research labs, colleges, all the institutions, they were closed. So we thought, being a researcher, we are always active. So we wanted how to utilize our time, and also to look into the research aspects, how we can contribute being a scientist to this pandemic, either as a medicinal chemist or as a any role. Uh, so we thought, can we start implementing the computational studies or computational approaches towards drug discovery to develop or to identify or to drug repurposing, you can say ideally. So that could be right term. So then we started, and I'm really very happy to share with you guys that we are now equipped with two of the potential molecules which are novel, newly designed in our, you can say, indigenous, and they have crossed the all preclinical parameters, and hopefully we will be taking them to next level of studies, that is clinical trials. So of course, let me start with drug discovery. Drug discovery is a very charming, very fascinating word, but it is not easy. We see that novel and effective, safe drugs are always in the requirement, because every drug has to lose their efficacy, or they compromise their efficacy by any mean. That could be either resistance or reduced efficacy, or maybe toxicity, because toxicity, that is the, it's a relative term. It needs to have a extensive study. Sometimes after 20 years of use, we find it that drug is not safe enough to use, so then we discard it. So I mean, drug discovery is not very easy, and it's a continuous process without any interruption. So it is something like that, kissing several frogs before you meet the prince. Now, why it is so? I tell you. So what conventionally what happens? You identify a disease. Either you are a biochemist or a chemist or a bioinformatician. Doesn't matter. So you first try to identify a disease, and then of course you go for target validation. Try to find what could be the possible target to understand the mode of action of that particular drug, and then hit identification, and then further structural optimization, that is also known as the lead optimization. Then there is the exit, which seems to be very easy, because if you see this barrier here, this barrier is this one: preclinical, clinical, and then marketing approval. All these things are not very much familiar or very much suitable for most of the academicians. Uh, there could be a lot of reasons behind that. Either lack of funding, or lack of support, or lack of infrastructure, many things. So what we do, we try to take exit from here. In turn, what we get? Good publications, repeat the process, and the life is over. So in whole academic, or you can say research life, you get hardly, maybe during this process you must have got lot of molecules or lot of drug, important therapeutic or potential candidates for an against any disease, but we have not looked into it. So my research is always, oriented towards this preclinical, clinical, and then to next. I don't know about how it is going to be, but yes, at least up to this preclinical, a researcher can take any molecule to this level. Of course, it involves a lot of hard work, a lot of efforts from every end, and collaborative job, and also a lot of funding. So that could be reason, because you can see there are like a few drugs which have been identified or developed from India. Particularly, I'm talking about synthetic drugs. So because this red portion remains always challenging and very tough for everyone. Now, there are various approaches which people have implemented globally. So the first term comes into is integrated drug discovery. So you see, drug integrated, when you say integration, I mean you have to merge, you have to collaborate, you have to come out of your comfort zone. Then only you will be able to contribute something. Either you consider your any infectious disease or other non-communicable disease, whatever diseases, regardless of any disease specificity, one has to be very open-minded if you want to work in drug discovery. You have to collaborate. You have to work with other people who work in different streams, maybe pharmacology, maybe other people who work in bioinformatics, let's say, who are very much into that. So then only you can lead your project. You can see you start, it's a drug discovery roadmap. So the simplest one is target validation. Of course, here you have to integrate with a right biologist, and then you go for hit generation. I mean the molecule which seems to be very good candidate. Then further it has to be optimized, because the compound or the molecule can have lot of side effects, though it is uh, giving very good activity, biological efficacy, but that can be compromised at any stage of life. So then lead optimization to understand or to bring one molecule that's going to be the best, and then further you go to the successfully deliver it to the preclinical candidates. Of course, it looks very simple, but you can see, you can realize that it is not at all very easy. For instance, malaria. Malaria is a, my main, focus of research, or is always, it was focused on malaria. So there also we have a few preclinical candidates, and I'm really very happy to share that we are in touch with the various pharmaceutical companies, and they have shown very deep interest, and hopefully we'll be getting their approval for next level of studies shortly. So for malaria, after 1996, you can, count the years. Now, after 1996, there is no effective drug, the single drug appear to the market. So now you can understand the real obstacle behind the drug discovery. So it can fail at any stage, even after 20 years of the development drug phase. Now, let me show you the timeline. So it is just a rough idea. The initial drug discovery screening assays that could take sometimes 5 to 6 years. For us, it took 10 years. uh, you bring to, let's say, preclinical. Out of 500 molecules, the clinical trial, if it reaches, it becomes just 10. You can see the success, ratio. And then further it goes to the next level of that is approval, FDA, either Indian or wherever it is possible. So ultimately it has to go to global market, so it has to go through the FDA review. So this whole process, you can now realize how difficult it is. You can just think the efforts as well as the uh, the cost of all this process. It is highly costly, but it is possible. So the possibility is just to collaborate with the right people at right time. Now, another approach what people have tried to implement, that is target based drug discovery. So I'll be coming to what people have done and what we have contributed using the computational approaches. That is the theme of today's talk. But before that, let me highlight few things what people ideally started following is that there is a plan which I have shown you up to the preclinical stages, but how it can be implemented practically. So of course you have to use any of the popular softwares which are available. So Schrodinger system is always available. Some of them, they are of freely available, but Schrodinger it has to be it's highly expensive. But we procured it in our lab. So then you try to screen the drug libraries, either FDA approved one or synthetic one or natural products. You can go to any category and then try to optimize the go no go plan of the study. Of course, it has to be first virtual screening and then go to biological assays, then lead optimization, whether it is going to be satisfactory or not. If it is not, just dump it and then again follow go no go and go to in vitro testing where you perform high throughput screening. Either it's COVID-19, since today's talk will be focused on COVID-19, so just I have highlighted it. Otherwise, you can do it for any disease. So there, if you procure your positive libraries, then again, you need to optimize its structure, that is lead optimization, using that has to be again aided with your computational approaches. And then further you go for the retrosynthesis. If it is, let's say, a known compound, or you have to optimize its synthetic procedure, and then again testing and cytotoxicity assays, and then your ultimately after your in vivo testing, it becomes a preclinical drug candidate. So it is really very complex process, but very exciting. 99% you will have failure, but doesn't matter. Even 1%, for keeping that in mind, that 1% will come, you keep on working that. And of course, it has to be supported with a lot of funding. So that becomes a very, very challenging barrier, at least in India. But fortunately, we are well equipped with funding, and even we have funds from abroad. So, okay, now let's come to the point. I hope we all are aware about the computer-aided drug design or drug discovery, and people call it as CADD, which is a very familiar term. So, why it is important? You can say it. So, ideally, if you look into drug discovery, it is conventionally, it used to be driven by the biological assays or a biologist. Let's say, for instance, I am a chemist, synthesize a library randomly, given to a biologist. They have screened it. I got the results, taken it. Okay, I considered it. And then accordingly, I have to modify the further my chemistry thing. But gradually, science has, grown up very fast, and technology together. So then the approaches have, they have modified so rapidly, and it has fastened the drug discovery process. So conventional approaches they have their own, advantages, but everyone wants to everything, to get in very short period of time, very quickly, but the best one. So keeping all those things in mind, probably people started using all these technology based approaches to fasten your drug discovery approach. So the design molecule is ideally targeted to have good interaction, binding affinities to the target protein or target enzyme, and it is anticipated that it will activate or inhibit the function of a biomolecule such as a protein. At the same time, you try to avoid or screen it accordingly using your CADD approach, that it will not have additional interaction with other unwanted proteins. So that's how you do it. So the objective of CADD is to avoid random screening against disease assays, and rational drug design and testing, speed up screening process, and target directed, that is de novo design, and integration of testing into design process, and also to uh, understand or realize that failed drugs fast. So let us say if you follow the conventional or traditional process and get the assays after synthesizing, let's say after a year you give the result, give the molecules to a biologist for the screening, or even a big pharma company, and you get the results, let's say after 1 year. So 2 years are waste, and none of the molecule is potential enough. So your 2 years, all efforts, money, everything gone waste. So all these things, points can be avoided using your CADD approach, and that's how you can say it has played a significant role to fasten your drug discovery process. Now, there could be a if you compare the conventional approaches versus your computational aided drug design, so it is time saving, cost saving, accuracy could be very high, and information about the disease you can without before going to the biological assays, you can procure a lot of information on that using your computational tools. And screening is reduced. You don't need to screen large number of molecule in biological assays, because biological assays, they are always very costly and time taking too, because it has to be done in triplicate at least to make sure that your drug is working or not. And then database screening, yes, that is very much essential. If you want to work, take some drugs from the databases which are already approved, or there are various database, zinc library for example, and then less manpower is required. So these are the advantages. Now altogether, all these advantages have contributed to the design or finding of a suitable molecule to treat, partly at least, the COVID-19 disease. We know the pain what we have gone through, not only the like in India, but globally, uh, the problems and the pathetic situation we had during last 2 years. So, of course, every the disease is caused by some agent. So, in this COVID-19, it is a SARS coronavirus 2, and the machinery of this SARS coronavirus 2 is partly, not completely, partly similar to the previously known SARS coronaviruses. And that's how it could be, it has contributed to understand the possible targets, what the main proteins which are responsible for its machinery to function. And that's how people have started targeting few proteins. You can see possible targets 1, 2, 3. I have 4 identified, highlighted it here. Target 1, that could spike proteins, but spike proteins, it is not a very good target, because it has always a problem of it changes its behavior morphology, and as a result, your drug may not be quite specific. But proteases and capping protein and helicase polymerase, of course, other targets, there seems to be very much potential. So initially, uh, there was a random screening under these pandemic situations. because there are some drugs which have failed previously for particular against particular diseases. So since but they were approved or they were in they have crossed the clinical trials for particular disease, so they can be reused. That is called drug repurposing. So in the beginning of this COVID-19, the global efforts were there to screen the already approved drug by FDA, and one of the very best example was remdesivir. But unfortunately, we have to be, very much honest with based of them on the basis of the data available. You see, the broad spectrum antiviral drug, of course, it was developed uh, by Glid Sciences from USA to cure the Ebola viral infection, but it failed because the compound, the drug is, it has a lot of adverse effects, hepatic or diarrhea, renal impairment, lack of available information contradicts our understanding of further adverse effects. So the drug failed against, and its efficacy is compromised by cytotoxic effects. Likewise, hydroxychloroquine, though drug is in use in India and other countries since long, almost 30, 40 years, but it was also tried and screened against, and it found that it is quite effective against to kill the virus. And other two drugs, lopinavir and ritonavir, they were also attempted, which are anti-HIV drugs, and they target the protease and main protease of the SARS coronavirus 2. But it has both the drugs, they also uh, try to show some adverse effects against liver functioning and gastrointestinal issues. So a variety of severity about side effects was shown by these two drugs. But remdesivir was shown to be the most efficacious drug, regardless of its toxicity. So it was in use everywhere. I think particularly in India, which was quite surprising for me at least. But anyways, under these conditions, one has to have something to treat the people when people are dying back to back. So it was probably essential to control the disease. And HCQ, that is hydroxychloroquine clinical trials, they were also quite successful, but it was banned by other countries. I don't know what could be the possible reason. Maybe there could be a lot of other reasons. So what we get out of it, the business benefits, global. So all those factors also matter in this business. So another, third drug that were in the combination of these two drugs, but there is no strong evidence for the efficacy of LPV treatment against COVID-19. Although there is increasing evidence that these two drug combinations showing promising results. So you can see now, although there are these all three drug, they are approved drug, but they have the adverse effects, and probably that was the reason that they were not taken to the use of public use. Probably, if I may be wrong, but what I know that the excessive use of remdesivir was there only in India, not in, I think, other countries. I may be wrong. Okay, now being a chemist or medicinal chemist, what we can do, how we could contribute to this pandemic situation. So we know that Professors Wolfgang always say that effective drug therapy ranks high among these scientific advances that have changed our lives over the past century. So I'm always inspired with this. And the next is the medicinal chemists play a crucial role in the drug discovery processes through the selection and synthesis of a variety of compounds, libraries, and that established SAR, that is structure activity relationships, and achieve efficacy and safety in preclinical animal testing. So these two quotes were significant enough to give a strong fire in us to work in this direction. And also, we have, we were working towards the discovery of oseltamivir analogs, and meanwhile we could, contribute a very nice impact factor paper that also supported us to look into this antiviral or beyond our ongoing efforts. So medicinal chemistry, of course, people say that I don't trust atoms, as I heard that they make up everything. So I take it very, very positive because it gives you everything. So the discovery or design of novel therapeutics and their translational into useful medicines, that is the main role of a medicinal chemist. Of course, it has to be substantiated significantly by other people who are in the process, either a biologist or a pharmacologist or other people who are there in the process. So design, synthesis, and SAR, as I told you, and receptor interaction, that is the target identification. And then ADME, that is cytotoxicity profile, safety profile of any molecule, that has to be established. And this work can be done by a medicinal chemist with the support of other people who are there to in the area. Now, let me tell you, this was our first effort in this direction using computational aided drug design, and we could take a library of molecules from the FDA approved or other zinc library and other lot of libraries we have screened and performed the high throughput virtual screening. And these were the 7 proteins we have taken, and they were subjected to high accuracy docking. And then their refinement was done, secondary scoring, that is GBSA, and then further the MD simulations to validate what we have achieved in initial molecular docking. And then top 10% molecules were further refined based on their high accuracy docking. And then further, at last, we have found a top lead molecules which were further tested for the viral targeting assays in the biological labs. So this is the presentation of presenting one molecule, that is bisindolyl melinimide, and the structure is shown here. The compound seems to be very potential against the replication of SARS coronavirus 2. And it targets it's a main protease, that is 3CL-Pro, and 3CL-Pro main protease is one of the essential protein, and therefore it represents as a potential target for drug discovery against SARS coronavirus 2. So these are the results, in silico results, docking results, and then MD simulation. The compound seems to be quite stable when it is complexed with the protein. And these are the biological assays result. You can see this is the molecule which we identified, and ivermectin. I think we all have heard this name, ivermectin, which is a broad spectrum antiparasitic drug. So this is in use everywhere. So this was taken as a positive control. And so this is entry blocking assays. So where, of course, our compound is not able to beat this ivermectin, but it is somewhere, at least it has some efficacy. Yes, this was our first attempt. So here it is, you can say 80% inhibition of the viral titer, and here it is 99. So somewhere the compound is able to compete. And overall here what we can see, the compound seems to be non-toxic within tested concentration days, and that is 50 micromolar. So overall, the results were compiled in collaborative job. Let me be clear with that. We tried a lot to get some biological assay support from Indian institutions, but unfortunately, there was not good cooperation. Probably at that time, only one lab, that is NIB Pune, they were able to screen. Later now, you can get the assays from, I think, even from INMAS and other laboratories, DHSTI. So, but at that time, it was very difficult to obtain the results from biological assays. So it was a collaborative work, and which could be published in a very good journal, Methods. And then since we found a one potential target, that is main protease 3CL Pro, then we further implemented the high throughput computational virtual screening, and we found that one of the molecule from our in-house library seems to be very potential against SARS coronavirus 2 main protease. So here, this is the crystal structure what people have reported, the main protease. And you see that these are three domain, and these are two histidine 41 and cysteine 145, two residues which seems to be very potential to have good interaction of any drug with these two uh, residues. And how your 3CL Pro protein contributes, and why it is so potential target, like one very simple question. So here it is justified. So you see, this is 3 CL Pro protein combines with other peptides and ultimately contributes to the formation or synthesis of another protein, that is NSP16, which seems to be a very essential protein. So if you block the activity of this particular 3CL Pro protein, certainly this NSP16 synthesis will be collapsed or will not work, or it will be inhibited. So then we thought, let us go in this direction further, and we performed a virtual screening of our in-house library of compounds. We have very good number of almost more than 10,000 library of compounds in-house, virtually designed. And here also indinavir was taken as a positive control, because at that time this was the potential molecule to be utilized. And our molecules, which were based on hydroxyethylamine, so they were screened, and out of 25 analogs, there were few compounds, and one of the compound is 16, which is shown here, which is a very simple compound and very easy to synthesize. So this is a docked complex with our compound with the main protease 3CL Pro, and you can see the results indinavir docking score, and it was quite competing with our molecule. And then further the favorable physicochemical or pharmacological properties of this molecule were quite convincing. And this shows the main role of a medicinal chemist, that is SAR analysis, which is again essential to look into the structure and why it is active. So we could see that there are two pockets. Pocket one, here it's a amine, and here it's a protected one. So this is the hit molecule structure, and which you can see these are other molecule. I have not shown other like potent molecules, because it will be quite confusing, and I don't want to focus on chemistry in this lecture at least. So the simulation quality parameters for this HEA complex for 100 nanosecond simulations in terms of total energy, potential energy, and pressure, temperature, and the volume, they were all these parameters were considered, and we could trace out that our compound, when it is complexed with 3CL protein, seems to be very potential, and it is highly stable. So that shows that our results are quite promising, and then when we wind it up together, and we could able to publish it in a very good journal, which seems to be one of the premier journal in this area. has impact factor 4.9, Journal of Chemical Information and Modeling from American Chemical Society. So this is the graphical representation of that overall crux of the molecule, crux of the study. Then in this direction, we further thought that how like we could approach the multiple proteins, because it's not a single protein, right? So it's a bunch of proteins which is able to catalyze the functioning of viral machinery. So then we thought, let us try to uh, implement our computational approach towards the COVID-19, and that was targeting multiple enzymes of this particular enzyme. So, which we anticipated that this could be a very potential approach to combat this disease. So, first protein was the protease, 3CL, this is known as NSP3 and PL-Pro protein, and another one NSP9, and then NSP12, which is also known as RDRP, which is the target of your remdesivir drug, people say, and NSP15, and that is also known as NEN2 enzyme. So these are four proteins we initially thought to target, and whether we can find a single molecule which is able to target these all four proteins. As a result, the whole machinery of the virus will be affected, and that may contribute to kill the virus, or at least to collapse its functioning. So this is the representation of our approach. You see the target proteins, these I already discussed, and then 1,520 compounds in-house, partly, and also we have taken various libraries, approved compounds. So there we could trace out that these are the four compounds: hesperidine and diosmine, and then catenolin and ascorbose. So these are the natural compounds. I think you all guys have the background of botany, so you must be very much familiar about this. So these are the four compounds which seems to be very potential and have very significant docking results, which were further confirmed by extensive molecular simulation studies at 100 nanoseconds. And there we could see that one of the molecule, that is diosmine, seems to be very much potential, and it is able to target all the proteins approximately in similar efficacy. So then these results were further confirmed by molecular dynamic simulations, and we could conclude that possibly based on our in silico results or computational results, this molecule, this drug candidate could be a very good inhibitor of all these four proteins, and thus its efficacy has to be confirmed in other biological assays. But unfortunately, at that time, it was not possible. Now we have submitted this molecule for additional biological assays. Probably we will be getting the results soon. So all these things were clubbed together, combined, and published in Methods, which seems to be another very good journal in this area. So next was a novel inhibitor of RNA-dependent RNA polymerase, that is RDRP, or you can say NSP12. So based on the information what was available at that time, because see, the virus was new, so nobody knew about it. Only you see the papers were appearing from China. Majority of the papers were from China, and they used to when they tried to publish the single crystal X-ray diffraction of data of those proteins, and then rest of the people globally, they started working on that. So of course we were on the same race, and we found that RDRP, they proposed that RDRP could be a very good target, Since your remdesivir triphosphate, which is a known drug, approved drug, uh, is also target. But let me inform you now, recently a paper appeared from very good journal that says RDRP does not target only, remdesivir does not target RDRP alone. It also targets the main protease, that is 3CL-Pro. So science is always growing, and it can be commented, or people can counter at any stage of growth. So here I see that the compound seems to be uh, our compound, what we screened, that compound number 1 here. It is the newly designed molecule, novel molecule, and we could see that this compound seems to be very good inhibitor of RDRP protein target, and we performed a very extensive computational studies. So these are the RMSD. I mean, it is your molecular simulation studies, dynamic simulations, and there we could see that our compound is quite stable when it is complexed with RDRP, up to 100 nanoseconds, and these results were clubbed together, and it was published recently in collaboration with the Martin's uncle, very good friend of mine from Czech Republic. Is a very good scientist. So then we got opportunity to screen our samples, our compounds, in against the virus in vitro. So we send all the samples to University of New Mexico and University of Loyola Chicago, where I usually go to have some assignments there, and we could see that our molecules, HEA, you can say HEA, whatever they are, they are our molecule, and we could see that one of the molecule, that is HEA61, seems to be very much potential. You see 60%. It's a toxicity data initial. So the compound does not seems to be toxic. At least it has 60% viable cells, and it has also very good potency against the uh, virus cells when they were treated with the SARS coronavirus 2. And we could trace out that the compound seems to be a very good inhibitor of RDRP, and this is the like prop like figure here. You can see the interactions, particular with the few uh, residues of the RDRP protein, and they said, complex enzyme, how it is packed inside the cavity and binding pocket. So it is very much effective based on these results, and we could further perform. Then we perform its synthesis. This is the calexin, what I showed you. So I don't want to discuss chemistry. It will go, but we just synthesize this molecule in our lab, and which we presumed that it could be a preclinical candidate based on the results what we had so far. And you see, this is a very interesting slide, that the calexinin, which is our own compound synthesized in Hansraj College, seems to be very, very potential when the results were compared with chloroquine. At that time, chloroquine, hydroxychloroquine, and even remdesivir, they were the main, choice global. So our compound seems to be 293 nanomolar IC50, 50% inhibitory concentration, and that seems to be really very good. None of the compounds showed, including the remdesivir, and no apparent toxicity to the human cells, primary cells, that is PBMC, and kidney cells, liver cells. So we have not shown the data here. Then we performed in vivo cytotoxicity, because that becomes one of the criteria. Okay, a drug could be very potential against virus, but what about cytotoxicity? So you just imagine, even remdesivir, hydroxychloroquine, they all, and leponavir, they all are facing problem of their side effects, their safety concern. So that's why probably they are not in public use. So subacute toxicity. So calexinin, that is our compound. It was injected to mice at 1200 mg per kg, which seems to be very high dose, but it was given in orally for consecutive days and left for 7 days, and we could see that the compound does not have cytotoxicity, as all the treated mice were healthy enough. And you see, then we performed the biochemical analysis of the blood serum, and you see all the parameters, they seems to be normal, nothing was abnormal. Further, we did histopathology of these mice, treated mice, and you see this is the control, liver, kidney, and stomach, and this is shown of our compound, that is calexinin at 1200 mg per kg, and you see there is no effect, or there is no difference between control and the treated mice based on histopathology. So at last we have this compound which was patented, and this is the structure of that particular compound, and in house library even more than 1000 analogs, high throughput virtual screening. So you can see how the computational drug design have contributed to reach this compound. So I cannot forget this journey of my life, and this molecule, and then testing, no cytotoxicity. Now you can compare our molecule with other compounds in pipeline. So drug candidates in trials, and this is on right side, this is our compound. So the poorly understood mechanism of action for those drugs which are in, majority of the drugs which are in clinical trial, and our novel mechanism of action target. And in that case, high dose requirement, our case is nanomolar activity, and efficacy is compromised with cytotoxicity. In our case, we could not see any apparent toxicity so far. And cross-resistant possibility here, unique substructure and target binding site, we anticipate that compound may not face the problem of resistance, because it does not target the spike proteins, where the possibility of resistance and mutation is very high. It targets the RDRP, possibly RDRP protein, so it may not face the similar kind of problem. And the complex secondary derivation from already expensive drugs. So these are basically derivatized drugs, and which involves complexity procedure for synthesis. But in our case, I can assure you that the compound involves very simple synthetic protocols, inexpensive reactants, and it can be scaled up very easily. So it was filed a US patent, a collaborative one, and the compound is now in vivo testing, and since it has safety profile is already established, so the compound seems to be very potential for next level of studies, and we hope that we'll be getting it the results soon. But of course, it is, as I showed you, that it is very lengthy process and very expensive, so we have to cross many barriers, being in small institutions, a lot of crisis, lot of problems, lot of obstacles, but I'm lucky to be in Hansraj College, so there are a lot of support from every end. Okay, so this was a little media coverage that we could see. Our things were highlighted in Times of India and other newspapers, print media, and also digital media. You can just have a look if you are interested to listen this.
और कोरोना वायरस की दवा बनाने की दिशा में एक अहम रिसर्च दिल्ली विश्वविद्यालय के प्रोफेसर ने की है। हंसराज कॉलेज के प्रोफेसर ने एक ऐसा मॉलिक्यूल बनाने का दावा किया है जो अब तक कोरोना वायरस के खिलाफ खासा कारगर रहा है। रविशंजन शुक्ला की रिपोर्ट।
दिल्ली विश्वविद्यालय में हंसराज कॉलेज के असिस्टेंट प्रोफेसर डॉक्टर ब्रजेश राठी दिन रात अपनी लैब में जुटे हैं। डॉक्टर राठी का दावा है कि इन्होंने एक मॉलिक्यूल तैयार किया है जो कोरोना वायरस के इलाज में कारगर रहा है और इसका कोई साइड इफेक्ट भी सामने नहीं आया है।
ये मॉलिक्यूल हम लोगों ने कंप्यूटेशनली डिजाइन किया था पहले, और हमने बहुत बड़ी लाइब्रेरी थी जिसको स्क्रीन किया था, और उसमें हमें पता लगा कि ये मॉलिक्यूल बहुत अच्छा हो सकता है। और इसमें अलग अलग, क्योंकि इसके टारगेट हैं जैसा हम देखते हैं, तो इसमें आरएनए पॉलीमरेजेस का टारगेट हमको लगा, और हमको लगा एक पोटेंशियल टारगेट हो सकता है, तो इसका आगे टेस्टिंग किया जाए। हमने लैब में सिंथेसाइज करने के बाद में इसको टेस्ट कराया, और ये जो मॉलिक्यूल है ये यहीं सिंथेसाइज हुआ हंसराज कॉलेज की लैब में, इसी में, और यहीं हमने सिंथेसाइज करके मॉलिक्यूल को बाहर भेजा। फिर हमारा जो कोलैबोरेशन, ऑफिशियल कोलैबोरेशन हमारा चलता है यूनिवर्सिटी ऑफ लॉयोला शिकागो वहाँ और यूनिवर्सिटी ऑफ न्यू मैक्सिको, कलेक्टिवली हम लोगों ने वहाँ पे इसका टेस्टिंग करवाया और जॉइंटली टेस्टिंग हुई है, और जो अभी तक के रुझान है बहुत ही इंटरेस्टिंग है। और उसी के साथ साथ इमीडिएटली हमने इसको, जो कि कंपाउंड को हम कैलेक्सिन नाम दे रहे हैं।
दरअसल हंसराज कॉलेज के साथ यूनिवर्सिटी ऑफ लॉयोला शिकागो और न्यू मैक्सिको यूनिवर्सिटी मिलकर कोरोना वायरस के खिलाफ दवा बनाने के मॉलिक्यूल पर रिसर्च कर रहे हैं। अब इस नए मॉलिक्यूल को अमेरिका में पेटेंट कराया गया है। फिलहाल इस मॉलिक्यूल का टेस्ट कोशिकाओं और जानवरों पर कारगर रहा है। लंदन की 2 दवा कंपनियां जल्द ही इंसानों पर ट्रायल करेंगी। अगर ये ट्रायल सफल रहा तो आने वाले दिनों में बड़े पैमाने पर दवा बनाई जाएंगी।
रेमडेसिविर है और हाइड्रोक्सीक्लोरोक्विन, जो कि रिपर्पस ड्रग है, बेसिकली उनकी बात करें तो उनसे अभी तक के रुझानों में हमारा कंपाउंड बहुत बहुत बेटर लगता है।
कोरोना वायरस के खिलाफ पूरी दुनिया में टीके और दवा पर रिसर्च हो रहा है। सीमित संसाधनों के बावजूद भारतीय वैज्ञानिक भी कारगर दवा और टीका बनाने के लिए रात दिन एक किए हुए हैं। नई दिल्ली से रविशंजन शुक्ला, एनडीटीवी इंडिया।
So now you can see that how the computational approaches and this all these protocols, how they have contributed not only to our lab, but globally. Pharmaceutical companies, Pfizer or Novartis, they have started using all these techniques long back. Even they have started gone to AI, that is artificial intelligence, that is also computational based thing, and machine learning. I'll discuss few slides on that also. So they have already started using it, and they have significantly contributed to the drug discovery area. So now, yes, my next slide is very, very important, and I'm really very happy to share that we have already started working on machine learning, and hopefully, since being a pure chemist, it's quite difficult for me, but I'm the person who does not leave anything so easily. So artificial intelligence and machine learning in drug discovery, these are the basically jack of all the trades. We know that why. So there are some questions, let's opening with the questions. Is a Pablo Picasso, he is a very great Spanish artist who is no more. He used to say that computers are useless. They can only give you answers. Another thing is, he is a humbly is a British mathematician and also entrepreneur, and he works on big data, which is again nowadays a very fast growing field. So data is the new oil. Now, impact of artificial intelligence and machine learning computational approaches, how these are impacting our daily life or the professional life. Let's say dynamic data analysis, healthcare, travel, e-commerce, social media marketing and insurance, acceleration, automation, self-driving cars, hazard handling, retail. So these are the applications which have been summarized here. You can just I think they all already discussed. So, but if you want to have more information on it, try to go to some nature and science. I'll share some papers. So that will help the students to go into this area. But this is a very fast growing area nowadays. Now, some key concepts. As molecular docking. So conventional molecular docking, what we have done so far. There is a program input to the computer, and then there is an outcome. But when you go for machine learning, molecular docking using machine learning, there is a swapping program and then output. So you can refine things accordingly. You can bring it to the accuracy, whatever you have, the program, and then provide data inputs, output, and that pairs together, and you ultimately you bring out the best results, and in a very short time with high accuracy over the conventional docking protocols. Of course, reinforcement learning and Bayesian learning, supervised learning, input output pairs, age, shoe size, height, weight, unsupervised learning, that is very important nowadays, output unknown, clustering, and these are other various programs, deep learning and neural network, and explicit knowledge programs, AI versus ML, machine learning, specific tasks, and AI, that's a thinking ability. Nowadays a lot of this AI approaches have come to practical. You can see in your car or in your other all computers and even mobile. So that is we have to give the credit to AI only. So, but unfortunately, this AI and machine learning, they are bulldozing its way through the years of wisdom. Now, how? ImageNet, and then Tesla Autopilot, and then AlphaGo beats world champ, and then deep fakes. That is the very much dangerous, and bots invent their own language, and AlphaFold. AlphaFold seems to be one of the very good and very fantastic, fascinating discovery of this. I'll show in the next slide how it is. Yes. Protein structure prediction from its sequence. So this seems to be very good achievement, and it is published in Nature last year. And the DeepMind's AlphaFold 2 algorithm was implemented, and that has significantly outperformed other teams at the Casp14 protein folding contest. So this was developed by Google, and they could see that's alpha folding, how it predicts. So maybe for more clarification, one can go to this paper. That's why I have written here source. So this is the first page of that particular paper. This is going to empower a new generation of molecular biologists to ask more advanced questions, of course. So it is a AI approach network developed by Google AI offshoot DeepMind. So one can go through this if they are interested. Okay, so AI also this machine learning and AI together, they also contribute to chemical synthesis, and they accelerate it so rapidly that can be just assumed MCTS, that is Monte Carlo tree search. That is the algorithm using the various policies, that's neural, and you see the time of screening 13 seconds per molecule. I mean, they can tell you whether you will be able to synthesize these molecule or not. Of course, that depends on the accuracy. What is the algorithm that you are going to use? So people try to keep on modifying it to acquire or to achieve the high accuracy. And of course, every technology, they have their own advantages and limitations. So if you see the advantages and limitations of AI and machine learning, The functional group tolerance was evaluated, feasibility of reactions evaluated via double-blind experiments, and invention of new reactions is now at the forefront. Of course, that's very fast-growing area in medicinal chemistry or synthetic chemistry. Of course, there are some drawbacks or maybe limitations which have not been overcome so far. Failed reactions are rarely reported while models require a balance of positives and decoys. So the stereochemical outcome, including enantiomeric or diastereomeric access, cannot be predicted. Let's not go to that much. Routes were not validated experimentally. So these are the few things. Maybe one can go through these two papers, which seem to be very, very interesting if they want to read about this, since it is pure chemistry. So I don't want to make you very much bored on it. So limitations, of course, this is the very good drug. Barenbrutinib is an orally administered, rapidly acting covalent drug molecule, and that's a tyrosine kinase inhibitor. It's being developed by Bristol Myers Squibb for the treatment of rheumatoid arthritis. So this was the drug that was developed by using your machine learning and AI approaches. You can see how fast it was. So it's very, very interesting to see, and one can go if somebody wants to go to this area bioinformatics. So these are the fast growing big data, AI and machine learning. These are the branches of this area which are emerging day by day and very, very interesting. They are never ending. It will be keep on growing. So discovery of this is another molecule which was discovered using these approaches. So this is again N-terminal kinase inhibitor, and so jack of all trades, as we showed. So artificial intelligence in drug molecule determination. You can see the chart that shows 3 to 4 years lead optimization, which was previously, I think, much higher than lead optimization, lead compound identification, secondary screening, 4.5 year, I think, which is reasonably okay, and preclinical 1 to 2 years, and then another thing is that your clinical development, that is 5 to 6 years. That is, of course, nothing can be done in that case. It takes its own way because it has to go to the human trial and a lot of obstacles there. So now let's come back to those questions. It's very interesting slide. So challenges and bottlenecks and way forward, what seems to be future for this. So these two AI and machine learning approaches. So eliminating the black box associated with AI ML model thought experiment. Structured data source and diversity of data. A molecular representation was a challenge which now seems to have improved significantly. Now, let's answer this. Data and AI are inseparable. Wherever there is data, there is AI. Answers are important. They are easy to come by once you have figured out the right question. So I think that makes sense. This slide, these questions. So this goes by both these great guys, and of course he's a very famous. Humbly is a very famous guy who works in big data. So now I was telling you about that Pfizer and Novartis and other companies, they have merged together and to use the AI and machine learning and the collaborative efforts. If somebody is really very much interested in learning this, so one should go through this paper. This seems to be a very good paper. And there they have shown or elaborated their approaches, how the collaborative work, all these pharmaceutical companies came together to bring out something, drug molecule, even against COVID-19, and machine learning and AI, how these two techniques have contributed, how computational techniques have contributed to this discovery of novel therapeutics against COVID-19. So of course, success builds on the collaboration with the right people. So dedicated team of medicinal chemists and cell pharmacology. Partnership is very important. So we have partnership with a lot of people, NII, Mayo Clinic, USA, University of California, University of Debrecen, University of Geneva, University of California. It has come again. So MIT, of course. So he's my mentor from MIT. I worked with him for a year, and that was the best part of my life, I can say, in science. So these are other guys. He's Dr. Agam, very good friend of mine from NII, and she is one of the very, very famous biochemists from University of Geneva, Professor Dominic, Professor Avidrosal, chair from Department of Medicine from Mayo Clinic, very good friend. And these are the very good. He's a very wonderful virologist from Fiocruz, Ministry of Health, Brazil, and Professor Enico Borbas from University of Debrecen, where I hold the position of adjunct professor for next 2 years. And he's another fantastic guy from Charles University Biosav institution from Czech Republic. That is the very good, institution. So these are two papers which appeared back to back from our group, and which have impact factor 12.94, which seems to be very prestigious journal. So this is the lab which we have in the campus of Hansraj College. You can see the name of the lab, Laboratory for Translational Chemistry. I mean, I'm not interested in synthesizing and designing. No, it has to go to the next level of studies. So translational chemistry, that is the objective, and we have a good team of 7 people in the lab. So these are the ongoing grants. Now recently we are, I'm really happy to share that BRICS, that is Brazil, Russia, India, and South Africa grant we got. We have not collaborated with Chinese people, and we got it. Being in college, it's very difficult to fight for such kind of grants, particularly international grants. And more importantly, there we are, leading. We are, coordinator for all the countries. So this is I conduct every year. I have to conduct a short-term PhD course and 4-week UG course on drug discovery at University of Debrecen, Hungary. So there's a big group, very nice people, and that is I had been KIPS fellow at Brazil. So these are the people who attended the course. And what did the thermometer said? That is the, at least I have a degree. So I have already made you, so much of bore, and thank you for your attention and the patience that you have, and thank you for the invitation. Thank you so much.
Thank you, Rathi. Wonderful presentation. I think I'm falling short of words to express my gratitude for such a wonderful presentation. You very aptly quoted Clive humbly, data is the new oil, and that is what you have done to keep the lamp of curiosity burning. You have ignited almost over 300 students who are there, 300 participants who were listening to you. So thank you once again. I would like to address some questions which I have received from the participants, if I have your permission.
Please.
Thank you. One is, what academic background would you suggest a student to go in for in case he or she wishes to opt for this type of research?
So, ma'am, first of all, thank you for your kind words, and it's my pleasure to be interacting with you all. And it's really very good when there is a seminars within the colleges, and we interact with our colleagues, which maybe probably I have never met you guys, but it is really very exciting for me, and very much happy and very much honored to be part of this. So yes, let me come to the question. So the question itself, a answer. So see, it does not require any specific thing. So whatever background you have, I worked. Let me give, because I always give my example because I know about myself. So I did my PhD on purely, it's a kind of you can say physical organic chemistry, just a lot of crystallography. But now once I submitted my thesis, I never seen back to my thesis. No, never just took my thesis back in my hands, and I had just chosen, maybe it's a north and south, something like that, the area which was of my interest. And let me tell you, globally, if you see, uh, people who are a very good physicists, they got a Nobel Prize in biology. So it is the time to not think about the streams, but think about the innovations, how they could be integrated, how they could be catalyzed. So regardless of any specific kind of the specialization, it's better to learn whatever you have been learning. Either you are a botanist, a chemist, or a other, let's say, zoologist, doesn't matter. What do you want to do? You will do it if you want to do. So in my case, I don't want to have such kind of boundaries and just always come out of the box. Suppose I am a chemist. Oh, no, no, no. I can just synthesize the molecule. I cannot think about beyond this. That means you are not a researcher. And that's what, ma'am, I learned from MIT people, that my boss, he just 3 months gave me the training just to come out of the box. Come out of the box. Don't be within your comfort zone. So otherwise, the research can be done at DU or any places, he said. So only thing is, the thinking is very important. So my perception, my advice would be that do the best and try to integrate, try to open your brain and try to have a good discussion with the people from different streams, and then you can design your own. It's research is not something goal. Research is a vision. Keep on traveling. Journey is very beautiful. Only thing is, and within this journey, I don't care about the destiny. What would be, what I will get? No. What? Let's enjoy the journey. That's my suggestion. Thank you.
Thank you. I think that is what new education policy is bringing about, integration and innovation. And I hope we succeed in it. How much time did you take to figure out the molecules that you have sent for field trials?
Oh, it took we were working on this molecule. That it's an in-house library of compounds. We were working almost since 10 years, and then in-house library was screened against using these computational techniques, and we could trace out that the molecule could be a good inhibitor of particular enzyme of this COVID-19, and so almost it's a 10-year job.
But COVID-19 came just in 2020.
Yeah, yeah. But
So,
so it was a in-house library. We were working on some other disease.
all right, okay.
So then, based on that, since we already acquired a lot of data on the compound, so then you can say it's a click. Like computational approaches have assisted us or supported us to change our direction. So from malaria, immediately our focus was then COVID-19. Oh, this molecule seems to be very potential. Then let's take it to the next level of the studies. Yeah.
Thank you so much. Another, I think this question is not from the student participant, but nevertheless, I'm asking, can I run Schrödinger system? Now, what is that? I myself don't know. He has asked, there's someone called Mr. or Dr. Shivam Kori, and he says, can I run Schrodinger system? What is that?
Schrodinger, it's a software.
That is what he has written.
So no, can I run my? I did not understand the question.
Can we give access to Dr. or Mr. Shivam Kori to pose his questions directly?
Shivam Kori can raise his hand. We will make his mic and camera work.
He has written, can I run Schrodinger system? And then it says Win 10, 4 GB RAM, 1 tab, Intel, whatever, 64-bit.
It has for this that can be easily answered, because see, all these softwares, they are very heavy, so they cannot be run using like on ordinary system. So we need to procure a very heavy system, and the Schrodinger people, they give the support. They try to give the specification also, and if he needs like all these specifications, he can just drop a message. I can forward all the details, whatever required to run this system. And they try to give a 1 month free trial, so he can just learn, no problem.
Thank you. I would like to inform all the participants that email IDs of all the resource persons are in the e-manual that we have circulated. So you can always get in touch with the resource person as and when you require. One question that was my inquisitiveness, maybe very basic. You mentioned indinavir, right? Some molecule is indinavir. What are the basis of naming a drug? Why did you do Indina? Did it have anything to do with the country's name or what?
People, a lot of criteria people follow. Like we named our molecule calixinin. So it binds with the calcium channels of the malaria parasite. So calcium. So from their calixinin, just we took it. So sometimes it's a choice of the inventor, and sometimes it's partly associated with the activity, its efficacy towards particular enzyme. So, and sometimes people try to give on their own name also. And indinavir, it's a very famous drug which is used. So like people keep on naming them based on various their priorities.
Thank you, Dr. Rathi. I think I would like to stop question answer session here, because after your lecture, we have Professor Suman Lakhanpal, Head Department of Botany's valedictory address at 11. So I take this opportunity to propose vote of thanks to our esteemed speaker. I, on behalf of the organizing committee, Department of Botany, Zakir Hussain College, would like to thank Dr. Brijesh Rathi. We are grateful to you, Dr. Rathi, for having consented to deliver the talk on computer-aided drug design, very relevant topic in the context of pandemic and the looming uncertainty towards treatment and prevention of the disease. Congratulations on being able to identify molecules which have been now forwarded for trials. I would also like to congratulate you for taking Delhi University to the global level, and that's a great sense of pride for all of us who belong to Delhi University. The other thing that you explained about role of computer-aided drug designing was really intriguing, and I think a lot of student participants have got excited about this new field of research. I think you also very implicitly explained why drugs against spike proteins may not be very successful, and proteases and helicases, which are helping the viral genome to replicate, offer better options for designing drugs, especially in case of COVID-19. We are grateful to you for having spared your valuable time in enlightening the participants about this field. We look forward to an offline lecture in times to come, and if you ever permit, we would like to take our students to your lab so that they can, feel closer to research. I would like to impress upon my young student participants that this workshop had all the very young speakers who have achieved so much so early in life. They are experts in their respective fields, and that should be a great motivation for all of you. Thank you, Dr. Rathi, once again. The college is indebted to you for a wonderful and illustrative lecture.
Thank you, ma'am. Thank you for your kind words, and thank you so much for the very nice opportunity. And it's pleasure is always mine to be associated with all the colleagues. Dr. Ruchi, I want to thank her because she contacted and we worked together in Venkateshwara College for some time. So thank you so much. And of course, certainly your students are always welcome. And no problem. Whoever is interested to even work for some time, we will be very happy to give them training in either case. Thank you so much.
I would also like to add one thing which just slipped my mind, and that was, all this time, whenever we did any conference or workshop, we always invited people from outside the university. And I think this was for the first time, most of the resource persons from inside the university, and that too from colleges, and that gave us great.
That is the very, very important thing that one should integrate, because if we do not try to integrate with each other, how we are going to integrate with the rest of the world?
Absolutely.
So that that approach is missing somewhere. So I really congratulate to all of you and thank you so much. Botany department has done a fantastic job. Thank you, ma'am. Thank you so much. May I leave now?
Thank you. Yeah. Dear participants, we take the program further, and as we had announced earlier, we shall be sharing the feedback form shortly with you for filling up and returning it to us ASAP. And this feedback form will be an important prerequisite together with your attendance record and assessment to fetch you e-certificate. However, we thought that we shall invite a few students for oral feedback, and a very quick oral feedback will be appreciated. Can we have Titiksha from Punjab University, Department of Botany, to give her oral feedback? Can you raise your hand? Titakshvi, Punjab University. Yeah. Can she be given the access to present oral feedback?
Yes, ma'am, we are working on it. We are making her a presenter.
All right. And the other person is Shikha from Deshbandhu College. Please be ready and raise your hand. In the meantime, you can come forward because very soon our speaker for the valedictory address will be here.
She has joined. Dr. Suman Lakhanpal has joined.
Yeah, I'll just take 2 minutes with her permission.
Yes.
Professor Suman Lakhanpal, if I have your permission to have feedback with 2 students.
Go ahead, Ratnam. Go ahead. Please take your own time. No problem.
Thank you very much.
Ma'am, Titakshi is a presenter now. She can switch on her camera and mic and she can speak whatever she wants to, like feedback.
Yes, Titakshi.
Hello, good morning, ma'am.
Good morning.
Am I audible?
Yes, you are.
Okay. So, good morning, one and all. Respected teachers, organizing team, and my fellow attendees. On the last day of this national workshop on bioinformatics, resources, tools, and its applications, I, Titikshvi Thakur from Department of Botany, Punjab University, would like to extend my heartfelt gratitude to each and everyone who has been a part and parcel of this workshop. This initiative by Zakir Hussain Delhi College has been immensely helpful in imbibing the practical as well as theoretical aspects of this interesting subject. The entire team has been working day and night to provide us with this opportunity to learn even in these unprecedented COVID times. Our keynote speakers provided all of us with a wider horizon for thinking, reasoning, and logic. Learning from our resource persons has been one of a kind experience as we got to learn from the best teachers at their work and that too from a pan India level. The hands on session made us actually perform exercises and all have gained much practical knowledge about this subject. All the knowledge imparted to us is invaluable. I would also like to thank the entire organizing team who very patiently dealt with us and helped us with all the issues that we faced at any instance. Thank you for being a wonderful support system. Also, the workshop had extremely inquisitive attendee students who made us think in different dimensions, and many new aspects of the topics were answered. I have really enjoyed this workshop and am looking forward to more such workshops. Thank you to one and all.
Thank you, Titakshi. Can we have now Shikha from Deshbandhu College?
Yes, she can speak. She's a presenter now.
Hello.
Yes.
Good morning, ma'am. Am I audible?
Yeah, you are audible.
Good morning to everyone, respected ma'am, and all the professors. I am Shikha Kumari, third year graduate from Botany Honours, Deshbandhu College, University of Delhi. I want to thank all the professors for bringing such a good opportunity for us. This course was very well organized and easy to understand, and everything was explained step by step, which helped me to grasp the concept very easily. Thank you so much, everyone, and especially the presentation parts and hands-on session. They are short, concise, and to the point. All the professors really make it very easy to use the tools of bioinformatics. Thanks to this workshop, now I want to take my master's degree in bioinformatics. Thank you so much, everyone. Looking forward for more sessions like this. Thank you.
Thank you, Shikha. Though there are many more people who want to speak their mind here, but now that our speaker for the valedictory session is here, I hand over the screen to Dr. Tabassum Jahan. And on my personal behalf, welcome Dr. Suman Lakhanpal for the workshop.
Thank you, Ratan. Thank you.
Thank you so much, Dr. Vatal. And a very good morning and a very warm welcome to everyone present here to this valedictory session of this workshop on bioinformatics. I'm very happy to share that we have today Professor Suman Lakhanpal with us for the valedictory address. She needs no introduction, but just to summarize things. Professor Suman Lakhanpal is currently head Department of Botany, University of Delhi. She has been in Delhi University from 2002 onwards. Before that, she served as senior scientist in NRC on DNA fingerprinting, NBPGR, PUSA. Her area of interest in teaching and research are plant molecular genetics, crop genetics, molecular analysis of biological diversity, and plant-insect endosymbiont interactions. She has supervised 11 doctoral theses to date and 9 MPhil theses, and 4 doctoral theses are underway. To her credit, she has more than 30 publications in national and international journal of repute and more than 20 publications as book and book chapters. She is actively engaged in research work with many projects which she has completed successfully, and many are still underway with national and international collaborations and fundings. She has been recipient of several awards and is active in disseminating the knowledge through workshops, training programs, and talks at various platforms. She She did her graduation, post-graduation and PhD from Delhi University, and then she joined Agricultural Research Services and she joined as scientist in CUSA NDPGR. And I'm really happy to share at this point that she is alumna of our college, Zakir Hussain, Delhi College, Botany Department. Welcome, ma'am. It gives me an immense pleasure to invite you for this valedictory address.
Thank you. Thank you, Tabasum. Am I audible?
Yes, ma'am.
Okay. Good morning, everybody. Dr. Ratnam Kaul, Dr. Malti, Dr. Babita, Dr. Tabassum.
Ma'am, your voice is not there.
Okay, now. Is it
Now.
okay now? Yeah,
Now
okay.
it's okay.
Okay. Good morning, everybody. Is it okay now? Am I audible now?
Yes, ma'am, it's perfect.
Okay. Dr. Ratnam Kaul, Dr. Malti, Dr. Babita, Dr. Tabassum Jahan, convener of the workshop, Dr. Ruchiveer, co-convener, Dr. Zishan, and other teachers of Botany Department, Zakir Hussain, Delhi College, and most importantly, dear participants. It's indeed a pleasure to be a part of this wonderful event. I was fortunate to be present in the last part of the talk by Dr. Brijesh Rathi. I can see how the speakers as well as participants are enjoying the workshop. So I'm really thankful to the organizers for giving me this opportunity to address the participants. My deepest appreciation for the organizers as well as the college, the principal and the college for organizing this extremely relevant workshop. And it's my privilege actually entirely to give a valedictory address on such an important topic. I'll be mostly addressing to the participants now onwards. Students, personally, I am quite envious of the youngsters for whom the bioinformatics is being taught at the graduation level, and I'm not sure, maybe it has already been introduced even at the school level by now. I say envious because we were introduced to the bioinformatics subject quite late. That means rather very recently in our scientific career. My contemporaries like Ratnam Kaul, and if Malti and Dr. Babita here will, if they have logged in, they will agree with me that we had not even in fact probably heard of the term bioinformatics in our almost entire education period. Do you agree, Ratnam?
Absolutely. It is a very recent field.
It's a very recent thing. So I really wonder if we really needed a subject like this or not. Arguably, I think we did, but it did not exist for us. And if it did not reach us, neither formally nor informally.
Yes.
So, dear participants, consider yourself really, really fortunate. Again, with an inaugural talk by none other than Dr. Professor Sudhir Kumar. Those of you who are just getting initiated, let me tell you, there could not have been a better initiation than this. A person of stature of Dr. Sudhir Kumar initiating you in this exciting area of bioinformatics is a very good omen for you, I would say. A wonderful talk where he catered the entire gamut of those interested in bioinformatics, starting from the beginners to the regular bioinformatician. Why I say that? Because he even explained very basics of things like bootstrapping, as well as he talked about his most recent contribution in this area, like little bootstrapping in real time. It was, that itself shows, the mastery of a person, the genius he is. So I'm saying you're very lucky to be attending this workshop. So, I'm sure you have learned a lot during the last 1 week or 8 to 10 days, and you have been provided with a very strong foundation in the subsequent lectures and hands-on practice sessions on various basic aspects of bioinformatics. Well, I'm not a bioinformatician. I will not be able to tell you something significantly more than what has already been told to you. I am essentially a biologist whose research needs intervention of bioinformatics off and on, that too not very regularly. I would like to touch some random aspects of bioinformatics which are relevant to a person who is interested in bioinformatics or who wants to take up bioinformatics in not much time. I'll not take more than 10 to 15 minutes. They are mostly the points which I wish somebody had told me when I needed to do a little bit of bioinformatics. Out of compulsion, rather, I would say, because it is required these days. The points that I feel are important, points that have bothered me from time to time. I feel bioinformatics, more than a subject, it's a tool. It's a toolkit. A professional bioinformatician is a tool maker. Somebody like Professor Sudhir Kumar, he's a tool maker. And we are at the other end. So it's very nicely placed, Dr. Sudhir Kumar in the beginning and I'm in the end, because he is the tool maker and you people are going to be, learning how to sharpen the tool. And we, particularly the senior people in science, are the end users of this tool. So what use is a tool set if there is no user? So we are the final consumers of this, very fast emerging and a very relevant subject. I'll be sharing my PowerPoint presentation with you, so please bear with me for a minute now onwards. Presentation.